Hi everyone. Today we are going to start with activity 7, which is the last activity in the nervous system, which is about synapses. One-way communication synapses. Page 76, 77. At the beginning, do you know what do you mean by synapse? Let's define a synapse. A synapse is a junction between two neighboring neurons called neuronal or between a neuron and an effector organ, muscle, which might be a muscle or a gland. Look at document B, it's showing you the synapse. You know that when the nervous message is going to move from one neuron to the second one, once it reaches the end of the first neuron, the message gonna be released through neurotransmitters from such a neuron to the second one or to the effector organ. This junction, the communication between these two neurons, we call it a synapse. So a synapse, it's a junction between a neuron and another neuron in order to transmit the nervous message, or between a neuron and an effector organ. So the synapse, when we talk about the synapse, what are the parts that include a synapse? Look here at this figure as well as at this figure. This is the first neuron, the neuron that is carrying the nervous message, through which the nervous message gonna move till it reaches its end. Such a neuron, we call it a presynaptic neuron. And at the end of the axon, yes, at the level of the terminal buds, we have synaptic vesicles, and inside the synaptic vesicles we have chemical substances called the neurotransmitters. Such chemical substances, they are responsible to transmit the nervous message to the second neuron. And the second neuron, the one that receives the nervous message, it is called postsynaptic neuron. So the synapse includes a presynaptic neuron, a postsynaptic neuron, and the space between such two neurons, it is called synaptic cleft. Look at the space in the dark orange color. The space between the two synaptic neurons, it is called synaptic cleft. And the transmission of the nervous message takes place by the action of neurotransmitters. Where do we find such neurotransmitters? They are found in the synaptic vesicles of the presynaptic neuron. So till now, we defined a synapse and we identified its different parts, which includes a presynaptic neuron, postsynaptic neuron, and synaptic vesicles containing neurotransmitters which are responsible for transmitting the nervous message, and the space between such two neurons, it is called synaptic cleft. Now, look at this figure. We are going to talk in details how does the transmission of the nervous message or of the action potential take place at the level of synapse. Such steps are very important. You might be asked about them in the exam or to list them to me when I ask you about the transmission of the nervous message at the level of the synapse. When the nervous message reaches the end of the presynaptic neuron, when it reaches the end, when the action potential arrives at the end of the presynaptic neuron, we have calcium channels along the membrane of the presynaptic neuron. The calcium channels are going to open and calcium is going to enter, calcium ions are going to enter from outside to inside, from the extracellular medium to intracellular medium. The movement of the calcium ions from outside to inside, it causes the fusion of the synaptic vesicles with the membrane of the presynaptic neuron. Once the fusion takes place, the synaptic vesicles are going to open, and look how the neurotransmitters are released into the synaptic cleft. Here, a very important phenomenon takes place. The opening of the synaptic vesicles and the release of the neurotransmitters is a phenomenon called exosynaptic. exocytosis. From its name, exocytosis, exo outside. It means the neurotransmitters are released outside the vesicles and they are released into the synaptic cleft. Then the neurotransmitters will bind to specific receptors found on the membrane of the postsynaptic neuron. And look at this square, it is showing you that the neurotransmitter is binding to the receptor which is found on the postsynaptic membrane. Once binding takes place, you can consider that the nervous message is sent to the second neuron and it propagated to the second neuron. But I'm going to ask you a question. Do you think the link, the binding between the neurotransmitter and the receptor is permanent? Is it permanent? What do I mean by permanent? Permanent, I mean, do they remain binding each other? Of course, no. The link between them is temporary, مؤقت. Once the message is propagated to the second neuron, the neurotransmitters, they are going to be hydrolyzed, okay, or degraded by a specific enzyme called hydrolase, and they are going to return back. They are going to be recaptured back by the synaptic vesicles for a new recycling, for a new nervous message. Okay, so these are the steps which are involved during the transmission of a nervous message at the level of synapse. Okay, look here, this is a neuron and this is another neuron, and these are the steps that are taking place at the level of synapse in order to transmit the nervous message from the presynaptic neuron to the postsynaptic neuron by the action of neurotransmitters. So from here you are able to know the role of the neurotransmitters. So they play an intermediate role between two synaptic neurons, which are the presynaptic and the postsynaptic, or between a neuron and an effector organ. Even if the message wants to reach the motor organ, the same steps are taking place because the neuron is going to send the message to the effector organ to react. So same steps are taking place. Here they are telling you when the neurotransmitter binds with a specific receptor site on the membrane, the nerve impulse is thus transmitted. I might ask you, when do we consider the nerve message or the nerve impulse or the nerve influx is transmitted? You tell me when binding between the neurotransmitters and the receptors of the postsynaptic neuron takes place, we can consider that the nervous message is transmitted. Then what happens for the neurotransmitters? A specific enzyme called hydrolase inactivates it, and the neurotransmitters will be recaptured again by the synaptic vesicles. Now let us talk about the function of the neurotransmitters. These neurotransmitters that transmit the nervous message, they have two functions. They might be excitatory or they might be inhibitory. What do you mean by excitatory? Excitatory, I mean by it that such a neurotransmitter might activate something or excites or induces, and it might be inhibitory. For example, it might inhibit something or it prevents or slows down. Okay, now you will be able to distinguish between them. Before talking about the functions of the neurotransmitters, here are some types of synapses. Look here please, look at the figure. Look here, the link is taking place between, yes, between an axon, here is the axon, and between a dendrite. Such synapse is called axodendritic synapse. What about here? It's taking a place between an axon and the cell body. The cell body, it has another name called soma. So such synapse is called axosomatic synapse. And here it is taking a place between an axon and the other axon. We call it axoaxonal or axoaxonic synapse. And look here, it's taking a place between a neuron, yes, or axon, yes, a neuron and muscle. We call it neuromuscular synapse, and it has another name, you have to know it, which is between a neuron and muscle. We call it motor end plate. What do we call it? We call it motor end plate, which is between a neuron and a muscle. These are the different types of synapses. Now, let's move to document C. Document C, in document C, we are going to talk about a neurotransmitter. You are responsible about it. It is very important. What do we have in document C? It is called the Lewis experiment. Let us check what was the experiment of Lewis about. In 1921, Otto Lewis made a historical experiment on two hearts of frogs that were only connected by a perfusion liquid. So he got two heart frogs, okay, and they are connected by a perfusion liquid or Ringer's solution. This solution, it is made of salts or ions to keep them functioning, okay. So such solution made of ions, it keeps the hearts functioning in order to check their work. What happens? Upon exciting the vagus nerve of heart A, he made an excitation for the vagus nerve. You have to know what do you mean by vagus nerve. The vagus nerve, it is the 10th cranial nerve. It is one of the cranial nerves. You know, cranial nerves, they are the nerves that are connected to the brain. The vagus nerve is one of them, is one of the cranial nerves. It is the 10th one. It is responsible for many functions, but now you will see it is responsible for what at the level of the heart. Once stimulation for the vagus nerve took place, what happened? We noticed that the heart decelerates until it stops. We noticed that the heart A, look, its speed decelerates, decreases until it stops, okay. After a short time, heart B also decelerates until it stops. After some time, yes, look at heart A. Once we make stimulation, the speed decreases and then it stops. We notice that then after it, heart B, it decelerates and the heartbeat stops. The solution received by B, this solution that was received by B, reveals the presence of kind of molecules called acetylcholine in neurotransmitter. They checked what happened. They noticed that since the connection between the two hearts only is the Ringer's solution, they noticed that, yes, there is a chemical substance, a neurotransmitter, okay, called acetylcholine found in the solution, here in the solution which was received by B. So what can we conclude from such experiment? Here they were able to know the role of the acetylcholine at the level of the heart. The vagus nerve, once it was stimulated, the heart A decelerates, the heartbeats of heart A decelerates, then it stops, and the same happened for heartbeat. We know here we can conclude that the vagus nerve, upon stimulation, it secretes acetylcholine, and acetylcholine at the level of the heart, its function was inhibitory because it decreases the heartbeats, then it stops them, inhibits them, and this was revealed by heart B because the same result appeared on heart B when acetylcholine passed from heart A to heart B through Ringer's solution. So here we were able to know the role of the vagus nerve as well as the role of acetylcholine at the level of the heart. So from document C, we concluded that the vagus nerve, which is the 10th cranial nerve, it has many functions. For example, it is responsible for the heart rate, gastrointestinal peristalsis, and muscle movements in the mouth, including speech. Ringer's solution, as I told you, it's a physiological medium made of salts to keep functioning normally. Now let's move to the function of acetylcholine. Acetylcholine, you have to know acetylcholine. So you are responsible about the main neurotransmitter, which is called acetylcholine. Acetylcholine, it's a neurotransmitter. it is responsible for, it performs two functions. It has an excitatory effect or excitatory function, and it has an inhibitory function. The inhibitory function was revealed by Lewis experiment, which is at the level of the cardiac level. It lowers the heart rate, right? So from document C, we concluded that the vagus nerve secretes acetylcholine, yes, which achieves an inhibitory function in the heart by lowering or decelerating the heart rate. And acetylcholine, you have to know its second function. It is responsible for all the contractions of the muscles. All the muscles, we are able to contract it by the function of acetylcholine. It has an excitatory effect at the neuromuscular junction by allowing the muscles to contract. So inside our body, acetylcholine achieves two functions: excitatory functions by allowing our muscles to contract, and inhibitory function, which is at the level of the heart by lowering the heart rate. And also you were able to conclude the role of the vagus nerve at the level of the heart. The vagus nerve, it decreases the heartbeats by secreting acetylcholine. Okay. Now let's move to document D. What do we have in document D? Document D, okay, we are talking about the function of acetylcholine. What do we have in document D? The effective excitation of the nerve fiber provokes the exocytosis of the synaptic vesicles, then the contraction of the muscles. They are telling you, when we perform an effective excitation on the axon, when the axon is subjected to an effective excitation, what happens is exocytosis. I mean by it, the vesicles gonna open and they are going to release the neurotransmitters, and muscular contraction will take place. But notice, if a direct excitation was applied on the muscle fiber, no exocytosis and no action potential is recorded. What do I mean by this? I mean by this that if the stimulation was, or the excitation was applied on the axon, yes, on the axon, the presynaptic neuron, okay, will make exocytosis where the synaptic vesicles are going to open and they are going to release the neurotransmitters into the synaptic cleft, and then the neurotransmitters are going to bind to the receptors of the postsynaptic neuron, which will lead to muscle contraction. But you have to know that when you receive an excitation directly on the muscle, directly on the motor organ, no need for the neurotransmitters to transmit the message. The message will be directly received by the muscles, so no exocytosis will take place and no action potential will be recorded by the oscilloscope, but the muscle is going to react immediately. Okay, so here we can conclude from here that the neurotransmitters, they act at the level of synapse. What do I mean by this? That when the stimulation is received by the axon, this will lead to exocytosis and release of neurotransmitters to transmit the message. But when the stimulation is applied directly on the motor organ, no need for the release of neurotransmitters because the motor organ is going to react immediately. Here I've done an interpret for such document. It's very important. We start by the result. There is exocytosis of vesicles and muscle contraction takes place after applying an effective stimulation on the nerve fiber. So the effective stimulus on the nerve fiber causes exocytosis of vesicles and thus muscle contraction, whereas no exocytosis and no action potential is recorded when direct stimulation is done on the muscle fiber and not on the nerve fiber. This is the difference. So when you receive the message on your muscle, it's going to react immediately, but when the stimulus is applied on the axon is going to undergo exocytosis to release the neurotransmitters and to send the message and to achieve muscle contraction. So direct stimulation on the muscle causes its contraction and doesn't cause the exocytosis of vesicles from the nerve fibers. Okay, so I mean by this that when you receive a stimulus on your muscle is going to react immediately. No need for the neurotransmitters to transmit the message. But when the stimulus is applied on the axon, it is necessary for exocytosis to take place and for the neurotransmitters to be transmitted, to be released, to transmit the nervous message to the effector organs in order to achieve contraction or any reaction. Now, in document E, we have many ideas. I divided it into 4 ideas. This is 1, here 2, 3, and the last one 4. Okay, let's explain each part alone. It's also about acetylcholine. So the main neurotransmitter you are responsible for it is acetylcholine, and now you will be able to identify another one called the curer. First of all, what do we have? Micro-injection of acetylcholine at the level of synapse, that's muscle contraction. They are telling you that when they inject it, yes, when they add it, what do I mean by at the level of synapse? If I asked you at the level of synapse, what do I mean by it? Where is it injected? At the level of synapse, it means in the synaptic cleft, okay? At the level of synapse, it is the space between the presynaptic neuron and the postsynaptic neuron. So when they tell you at the level of synapse, we mean by it at the synaptic cleft. Micro-injection of acetylcholine at the level of synapse. So when they injected acetylcholine at the level of synapse or in the synaptic cleft, there was muscular contraction. How can you explain this? It's very simple. This means that acetylcholine, they moved from the synaptic cleft and they bind to the receptors of the postsynaptic neuron, and once they bind, the message is received and it causes muscle contraction. So what can you conclude from such experiment? Can acetylcholine works at the level of synapse? Can it work at the level of synapse, which is in the synaptic cleft? Of course, yes. So if the neurotransmitter is added in the synaptic cleft, it will work normally because its main function is at the level of synapse. So as the conclusion, so acetylcholine is the neurotransmitter that causes muscular contraction. The second idea, the increase in the dose of acetylcholine leads to an increase in muscle contraction. You have to know also that we have a directly proportional relation between the amount of acetylcholine and the muscular contraction. The more we have acetylcholine, the more your muscles are going to contract. So as the concentration of acetylcholine increases, the muscles are going to contract more. So they are directly proportional to each other. So the reaction of the postsynaptic neuron or the effector organ, it is directly proportional to the concentration of the neurotransmitter, okay? Whatever the neurotransmitter it was, not necessarily acetylcholine, okay? So the concentration of the neurotransmitter, it is directly proportional to the reaction of the effector organ or of the postsynaptic neuron, okay? Second, what do we have? 3, they told you, yes, autoradiography method is used where labeled the precursors of acetylcholine shows the presence of the radioactive acetylcholine on the postsynaptic membrane. They inject, what do I mean by inject? Added. They added radioactive precursors of acetylcholine. You know that acetylcholine, the main neurotransmitter, it is made of precursors. Okay, at the beginning they are precursors. They added the precursors of acetylcholine in the presynaptic neuron. So in the presynaptic neuron, they added the precursors of acetylcholine. Do you think they are going to act normally? Of course, yes. Then they told you that they apply a stimulus. Once they added precursors in the presynaptic neuron, and once they apply an effective stimulation, they noticed that the radioactivity appeared on the postsynaptic membrane. How can you explain this? It's very simple. This means that once stimulation took place, the precursors of acetylcholine, they undergo exocytosis, where they open the synaptic vesicles and they release them into the synaptic cleft, and then they bind to the specific receptors that are found on the postsynaptic membrane, and they cause muscular contraction. So here they are showing you, how did we know this is the result? Because the radioactivity appeared on the postsynaptic membrane. Okay, so this means that they have moved. So this means that the message have moved from the presynaptic neuron to the synaptic cleft to the postsynaptic membrane, and it is transmitted. We followed the direction of the radioactivity. Okay, so this experiment verifies that acetylcholine is the neurotransmitter at the synapse. This means that it works at the level of synapse. What do I mean by this? That, well, whatever happens, acetylcholine, if they are injected in the synapse, in the synaptic cleft, or if they are if you are subjected to a stimulus, acetylcholine are going to be released into the synaptic cleft, and they are going to bind to the receptors, and they are going to cause muscular contraction. Okay, so yes, they act at the level of synapse. Finally, here you are going to identify a new neurotransmitter. It is called the curer. Curer, here let us check out the function of curer. Look at this experiment, you will be able to know its function. Injection of curer in the synaptic cleft followed by nerve fiber stimulation, thus no muscle contraction paralysis. They are telling you that when they added a curer in the synaptic cleft, and when they applied a stimulation for the axon, okay, there was no muscle contraction. What do I mean by this? I mean by this that once a curer was added in the synaptic cleft, and once the axon was stimulated, once it will be stimulated, the synaptic vesicles are going to release acetylcholine, right? And acetylcholine causes muscular contraction. But we noticed that there was no muscle contraction, there was paralysis. Why there was no muscular? What prevents acetylcholine from causing muscular contraction? Actually, it is curer. Curer, it has an opposite function to acetylcholine. It prevents acetylcholine from making a contraction. So what happened in details is that once curer were added, they were able to bind to the receptors on the postsynaptic neuron, okay, of acetylcholine, and they stopped muscular contraction. Why they have the ability to bind to the receptors of acetylcholine? They have a characteristic, you have to know it, that acetylcholine and the curer, they have the same configuration or the same shape. So as a curer, since it has the same shape as acetylcholine, it has the ability to bind to the receptors of acetylcholine that are found on the postsynaptic membrane, and by this they inhibit muscular contraction and don't allow the muscles to contract, but they cause paralysis, shalal. Okay, so there is no muscle contraction when a curer is injected in the cleft followed by nerve fiber stimulation. So curer actually inhibits muscle contraction. So till now, you identified the function of acetylcholine. It causes muscular contraction, but curer, it has an opposite function. It inhibits muscular contraction, and by this we say that curer and acetylcholine, they are antagonistic. What do you mean by antagonistic? They have opposite functions. Okay, they work in an opposite manner. Acetylcholine causes muscle contraction, but a curer prevents muscular contraction. How does this take place? Since a curer blocks the receptors of acetylcholine because it has the same conformation or structure, and by this it inhibits the action of acetylcholine on the muscle by inhibiting its contraction. So that's it for experiment 4. Thank you. This is all for the activity of synapse. Any question, please ask me, and copy all the slides on your notebook because it's very important. Thank you, my lovely students.